Glycogenolysis is a process by which glycogen molecules in our body are broken down into glucose units. First, the enzyme glycogen phosphorylase removes the terminal glucose residues as glucose 1 phosphate from the non reducing end of the glycogen molecule. Glycogen phosphorylase enzyme is a dimer having identical subunits, and it attacks the exoglycosidic bonds. Pyridoxal phosphate is an essential cofactor in the glycogen phosphorylase reaction, and it is attached to the lysine amino acid of the enzyme. The phosphate group of the pyridoxal phosphate acts as a general acid catalyst, and it promotes the attack by an inorganic phosphate molecule on the glycosidic bond. Glycogen phosphorylase acts repetitively on the non-reducing end of the glycogen molecule, removing glucose residues until it reaches 4 glucose residues away from alpha 1,6 branch point. At this point, glycogen phosphorylase cannot carry out the process, and further degradation of the glycogen molecule is carried out by another enzyme called glycogen debranching enzyme. This enzyme, also known as glucan transferase, has two different activities. First, the transferase activity, in which the enzyme removes the terminal 3 of remaining glucose residues and transfers this trisaccharide moiety intact to the non-reducing end of another branch. Second, the remaining glucose residue, which is still attached to the main chain, is removed by an alpha 1,6 glucosidase activity of the same debranching enzyme. Thus, about 92% of glycogen's glucose residues are removed as glucose 1 phosphate, and the remaining about 8%, those at the branch points, are released as glucose molecules.